Hello everyone. So today I want to go through a worksheet. I'm going to post this worksheet onto the class room, but I'd like to go through this worksheet together because I think that it's a really valuable way of looking at the DNA replication that we talked about in our last video. And DNA replication can be a really complicated idea, so we're just going to walk us through some of the steps in that DNA replication. So in this little practice, we're going to use a couple different colors here. I'm going to use blue for my original DNA strand. I'm going to do the leading strand in red, and I'll try to do the lagging strand in a green here, just so you can kind of keep track of which one is which. And I'm probably going to add a little bit more detail than they are going to ask for. So in this first section over here, they say draw a DNA double helix with a sequence on the 5' to 3' strand of ACCGTATTGATC. So this is just to give us something to start with. So I'm just going to draw my DNA. So what I want to think about in DNA is that DNA is double-stranded, unlike RNA, which we really haven't talked about much yet. And then on this strand, we're going to draw in the 5' to 3' direction. So I'm going to put 5' down here, and I'm hoping I won't regret that. And then I'm going to put and then on the other side, because it's anti-parallel, then that means that the other side must be in the opposite direction. So remember, these 5 to 3' is just there to give us an orientation, tell us what direction we need to read this sequence, because sequences are incredibly important in DNA. So there's going to be a whole bunch of DNA nucleotides down here, blah, 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 right? An equal number on each side. I guess I added one. So maybe actually let's draw these out as sort of bars right across. So we've got a bunch of DNA strands like this, or nucleotides that go across. And then at some point our sequence here starts that we care about. So this is the sequence where we're going to care about. I got an A on this side, so that means I'm going to have a T on my opposite strand. I'm going to have A, C, C, G, T, A, T, T, G, A, T, C. So that's the one strand, and it goes in the 5 to 3' direction there. And then on the other strand, I'll have a complementary sequence. So T, G, G, C, A, T, A, C, and then G. Oh, nope. A mutation. Okay, T, A, and G. And then, of course, the DNA strand would keep going, and there'd be more bases. And I don't know what those letters are, but we're going to keep track of this little section of it as we go. So the next thing is that helicase begins to unwind these strands. So I guess actually at a different point down the DNA strand, right, somewhere down here is going to be our replication, our origin of replication. And with that origin of replication, we'll also see the replication complex will be down there somewhere. So we're going to say that's down there, way down the DNA strand. And now helicase is starting to open up the strand. So I'm going to just copy my strand out here, and I'm going to do a little bit of editing. So over here, we're talking about the same strand of DNA, only something's going to be happening here. We're going to have our original strand like this, but there's going to be helicase here, and helicase is going to be unzippering the strand. It'll be pulling these bases that we so carefully drew out here. They'll be being pulled apart to the opposite sides of the strand, and whatever these other fantasy bases are, they will just be pulled out to the side as well. Something like that. And of course, along this outside edge, we would imagine that there are going to be the single-stranded binding proteins, SSBs, and those will be on the sides holding the thing open. And in the meantime, and I'm going to do all my enzymes in highlighter here, this will be my helicase. So it'll be my helicase. So here's helicase, and it's going to be unwinding the strand. Now farther up this DNA strand, ahead of the helicase, is actually going to be that topoisomerase. Will be a little bit farther up, and it's going to be taking off some of that torsion. So that's not the most important detail of this process. That's why sometimes I don't talk about it even. Some years I didn't mention it at all. But this is the topoisomerase. And in fact, it's probably like maybe tens, maybe dozens, maybe even hundreds of bases ahead. And it's way up ahead, and it's unwinding the DNA strand. It's not unwinding, but it's cutting the DNA strand, flipping it twice, and then sealing it back together to remove any torsion that's building while this helicase is trying to pull the strand apart. And we have these single-strand binding proteins, which I did mention I would put these in highlighter. So let's put those in highlighter so that they are kind of highlighted. So that's their single-strand binding. So now we have a replication fork. So let's call this the replication fork. Previously, the replication fork would have been down here as well, right? Replication fork would have been way down there, and now the helicase is here, and it's starting to move this direction. It's been moving this direction the whole time, but in our first image, we couldn't see it because it was off the page. All right, so DNA polymerase adds complementary bases in the 5' to 3' direction to form the leading strand. Okay, so I'm going to need a copy of this image here. I've drawn a lot of stuff on this image. It's going to get a little bit messy. Let me see if I can pop that in there. Copy. Paste it a little bit off to the side so I can clean it up a little bit. Clean up this. Okay, so I'm going to move my helicase up a little bit so that I can open my fork up a little bit. And so let's see, let's get rid of that one, and let's open it up a little bit. So grab my original strand, I'm going to pull it open like this, and I'm going to pull it more like this. Okay. And now I'm going to move all my things over so they're back on the side. And what's going to be happening here is that the DNA polymerase, and I think I'm going to probably end up having to lose some of these up because it's not going to fit, but that's okay. Okay, we're going to try to jam that in there. Maybe we'll shrink it down so it fits. Okay, so the DNA polymerase now is adding complementary bases in the 5' to 3' direction to form the leading strand. All right, so this polymerase that we're going to draw as another green enzyme here. This polymerase, this is going to be our poly 3, is coming up the strand, and it is adding complementary bases in the 5' to 3' direction. So what this means is that it is making the new strand right here, and this new strand that is forming is going to be forming in that 5' to 3' direction because it will be complementary, right? So it's going to be complementary to the strand that is already there. And what did I say? I said leading strand was going to be red. Okay, good. So there's my leading strand. So that's my leading strand, and I'm going to put my helicase back. My helicase was right here, and the polymerase is going to be adding these bases. So these bases are going to be formed continuously in that direction. Now the DNA polymerase will add complementary bases on the other strand as well, And for that, we're just really going to take the whole picture here, I think. And I think we can actually just use the whole thing and put it over here. So just to emphasize that other strand, we're going to have another DNA polymerase on this side, but this one is moving in the opposite direction. So what color did I say I was going to make this strand? A green strand. Okay, so what's going to be different on the other side is that there should already be some DNA over here. This is also 5' 3' DNA. So it was made in this direction, just as this red one was made in that direction. So made in the same direction, but was made earlier, and looks kind of like this. So that's the one piece, and then here is our new chunk that would have been made by this polymerase that I just drew. So I'm going to draw it here. And so here's my new piece that goes here. But when this DNA polymerase meets up with this chunk that was already made, it's just going to fall off. It's just going to fall off. It says, hey, there's another chunk here. I don't do anything with I don't do jumps very well. I can't do the break. I can't jump this loop here, so I'm just going to fall off and go away. So this polymerization is happening in this direction, while the leading strand is happening in the same direction that the helicase. Remember, the helicase is moving this way, and the polymerase is moving this way. So that's why this is the leading, because it's following the helicase, while this one over here is our lagging strand. And so now we have an Okazaki fragment. This is we've run out of space to write here, but this is our Okazaki fragment. It's not connected to, there's no connection here, and there's no connection there. So it's a fragment. It's separate from the rest. And so now I'm going to need to, and so I guess we'll get rid of this polymerase, and we'll introduce the new enzyme we need, which is the one that's going to heal that spot, that broken spot, and that is going to be the DNA ligase. So the ligase will come in and it'll fix that little breakage in the strand, and there'll be a couple other things. We did miss the one step we missed was the DNA primase that should have come in to set up this whole process of making the Okazaki fragment, but you know that takes a lot of that is very difficult to draw out. So at the end of the day, what we really want to do is we want to take this whole thing here, and we want to copy that. And we're going to write that over here on the other side in the last step, except we're going to take this whole section here, and we're going to split it off. And I might have to move this one out too. And so for this little tiny section of DNA, what we should be seeing is a leading strand will essentially have extended all the way along this side, and as that leading strand was extended, I'm going to draw the bases in the order they would have been written. They would have been written in this order by the poly 3. Okay, so as the poly 3 read the what's known as a template strand. The original strand is often labeled as a template strand because it's what the strand is built on. And then on the other side, we would have had little sort of sections. Actually, I should write them out properly, shouldn't I? So little sections of the DNA would have been made one at a time, and would have completed this bar here. However many I was missing. So, you know, the first Okazaki fragment might have been like this. Then maybe there was another Okazaki fragment. Now, these Okazaki fragments would actually be like hundreds of bases long. So, almost impossible to draw on a paper diagram like these. So we do our best, but you know, we do have to deal with the limitations of how much can I really make you draw in, you know, in the time of a class, for example. So okay, so there's another G, and oh, I should actually, I'm not writing these in the right order, am I? Okay, so let's think about how they would have been written. They would have been written backwards. They would have been written like this: A T A C G G. And then that would have been a fragment, and then ligase would have come along and done the little bit of gluing there to fix that. Again, we would have had another strand, another Okazaki fragment form on this side, and it would have been spaced away. It would have formed the bars at the top here, whatever bases those were, then added the guanine, the adenine, the thymine, the cytosine, and then it would have gotten to this gap, and it would have been stuck at the gap and would have to wait till DNA ligase came by and said, well, for starters, you didn't really align those very nicely, John. And then DNA ligase would come in and put in a little bit of glue there. And so in that way, that's how we would end up with the two strands. We can see it's semi-conservative because each strand has one blue bar from the original and one bar that's from the new one. One of the strands was formed with lagging, and one of the was formed with leading. Now, I do want to stress that they were not perfectly formed from leading and lagging, right? Because there are many origins of replication happening all across the DNA strand. So those origins of replication would have resulted in many places where leading and lagging were started, and leading and lagging strands will collide with each other at some point, and DNA ligase will have to get involved to connect those two strands. Somewhere way up here, there's a leading strand that's formed that's coming in this direction, and then somewhere down here, there's another leading strand. There's a lagging strand somewhere that they're going to come into contact with, because each of those, think about this, that each of those little origins of replication look like this, and on one side, let's call this a 3', 5'. So that's maybe I should use blue, so it's the original. So think about this. Here's an original strand, right? And it looks like that, where the origin of replication has opened up, 5', 3', but the origin is a point that starts. So then from that point, if you are looking this direction, then you would see this strand as being 5 to 3. So 5 to 3 would be the strand on the left here. So we would see that one be formed by lagging. And on the bottom, you would see, looking in this direction, you'll see a 3 to 5. So that means that it would be formed as a leading strand. But looking in this direction, we can see that we are at the 3 part and we're looking towards the 5. So this side would form continuously, and this other side would form discontinuously, because we are going to have 2 of these helicases, one on each side, moving in opposite directions. So in that way, neither strand is perfectly made of leading strands or perfectly made of lagging strands. It's actually always going to be a combination of the two that have an effect. All right, I think that's good for now. This worksheet in particular has another section to it, but I like this diagram. Try to do this diagram at home. You can just do it on a piece of loose leaf and see if you can work your way through it. That would be my recommendation. I'll post the blank, and I'll post this filled in one, and I'll let you have some fun with that. All right, we'll talk to you next time when we talk about transcription.